Can, hello, hi. I'm Dr. Anthony Marrota, Chief Scientific Officer from Augurex Life Sciences Corp. And today I'll be presenting data on soluble 14-3 eta, focusing on a personalized rheumatoid arthritis patient management strategy. These are my disclosures. And these are the learning objectives for the particular presentation that I'll be giving today. So one of the first things that I think is important as a learning objective is to describe how 14-3 eta, specifically in the context of RA, differs from the other 14-3 isoforms. As part of the learning objective, we'll also look at 3 different ways in which 14-3 eta informs clinical management of RA. We'll talk a little bit about the 4 biomarkers that have resulted as a consequence of our R&D efforts, and then we'll explain or look at why 14-3 represents a rational drug target. Before I get into the presentation, I think it's important to delineate how Augurex came to fruition. As a scientist working in the reagent space, we realized that there were a lot of early stage technologies at the level of the university that looked very promising, but really didn't have the robustness around the data to garner the support or attention of the large diagnostic companies. So one of the things that we elected to do was to really create a company that would fulfill that gap, where we would take these early stage promising technologies, fulfill the middle piece by really working with key opinion leaders to understand not only the unmet needs, but to advance the marker along those lines, all of which to get to the point that these markers would look attractive to the larger commercial diagnostic partners. And what's nice is that we fulfilled this model working with US partners, and we're currently working on advancing our marker in the rest of the world. So we've recently obtained Health Canada approval, European approval, and so now it's nice to be at a point that the marker can actually be commercialized and used clinically. So our founding technology is the 14-3 eta protein, and this discovery actually came about through the University of British Columbia as well as the University of Alberta. There was a scientist working in the area of skin and wound healing, and he made an interesting observation that 14-3 sigma, one of the other isoforms, 14-3 isoforms, was actually released in through dermal fibroblasts, and when it was released through dermal fibroblasts, it would feed back onto the cells, stimulating them to produce MMP1. Now, in RA, one of the major issues is the synovial fibroblasts. They actually become very active, and they form what you would consider a non-malignant growth, almost like a tumor. And these fibroblasts are intimately linked to joint degradation. And so with that, one of the things that the second discoverer, who's a rheumatologist, initiated was, well, if 14-3 sigma in the context of skin and wound healing is playing a role in the induction of MMPs, I wonder if any of the other 14-3 isoforms play a role in the context of RA and could contribute to the joint damage process, since MMPs are linked to joint degradation. So with that, the 2 investigators, the basic scientist and the rheumatologist, decided that they would screen synovial fluid and serum from patients with inflammatory arthritis to look to see if any of the 14-3 isoforms were relevant. And so what they did, so SF on the figure here represents synovial fluid, PS is matched patient serum, and K is a cell lysate. It's a keratinocyte cell lysate. So based on the profiling of the different 14-3 family members, what they saw was that 14-3 eta as well as gamma were detectable in both synovial fluid and matched patient serum, and that the levels of the markers, so the 14-3 eta and gamma, were significantly higher in patients with inflammatory arthritis than in corresponding healthy controls. They also observed and reported in a paper that the levels, the 14-3 ADA levels were 5 times higher in synovial fluid than in matched patient serum. And incidentally, one of the other observations was that 14-3 ADA was significantly higher than 14-3 gamma. So through this, it was based on this technology that we essentially licensed in 14-3 ADA and spent the last 5 years advancing it to the point that it's clinically ready. So before we get into some of the clinical data, I think it's important to understand what the 14-3 proteins are and why 14-3 ADA is so relevant in the context of RA. Now, the 14-3 proteins, they've been discovered approximately 50 years ago. There's a lot of interest, obviously, in terms of their intracellular function. They behave as molecular chaperones, regulating an array of biological processes. They were initially discovered as brain proteins, and there's evidence that in the context of certain neurological disorders, that other 14-3 isoforms have utility as a diagnostic marker. So for instance, 14-3 ADA is used to diagnose Creutzfeldt-Jakob. Now, they were coined as being, or named as being 14-3 proteins based on how they eluted during purification. There are 7 different family members sharing approximately 50% amino acid homology, and the relevant isoform in RA based on the initial discovery as well as all of our data is 14-3 ADA. So from this point forward, I'm going to focus on 14-3 ADA. I should mention too, I apologize for this, that if you have any questions, please feel free to send through your questions and I will address those at the end of the talk. Now, the 14-3 ADA protein itself maps to chromosome 22. Its size is 246 amino acids in length, giving it a molecular weight of approximately 28 kilodaltons. And when you look at 14-3 ADA across different species, it's highly conserved. So when we look at the human, mouse, and rat, there's only 2 amino acid differences, which are denoted on the slide in red. So they're highly homologous. Now, when we look at the 14-3 protein more structurally, it's a small acidic protein. It consists of 9 alpha helices, and it has a variable carboxyl and terminal end. When they come together, 14-3 proteins as a whole can either homodimerize, so they bind to one another, or they can heterodimerize. And through that dimerization process, you can see on the image on this particular slide that they form an amphipathic groove, which allows them to bind with various ligands. Now, a lot of the data shows that 14-3 proteins as a whole bind to distinct phosphopeptide moieties, but there is recent evidence as well that the 14-3 proteins can bind to other particular sites that are independent of phosphorylation, hence they don't require phosphorylation. So it's through these phospho and non-phospho binding motifs that 14-3 is able to interact with various ligands. And to date, there's over 200 ligands that the 14-3 proteins bind to, and for 14-3 ADA, there's approximately 150. Now, when we look back to 14-3 ADA and RA, one of the other things that the investigators realized through their efforts was that when they looked at 14-3 ADA and related it to MMP1 expression, they found that there was a very strong correlation between the levels of 14-3 ADA in synovial fluid and serum with that of MMP1 and MMP3. So if you look, the correlation between 14-3 ADA and MMP1 was 0.83, and with MMP3 it's 0.77, and that correlation held true and very significant also in serum. So it was with this that the investigators thought that 14-3 ADA might actually contribute to the joint damage process by upregulating MMP1 and MMP3. So as part of that, the investigators also performed stimulation studies, and what they found was that 14-3 had soluble ligand-like properties and was capable of inducing factors like MMP1. So again, it was really very much with this preliminary data that we decided to license 1433 in from the university and really advance it. Now, when you look at RA, it's not unlike cancer. It's very important to find patients early, to know who's at risk of developing more aggressive disease, and to have markers that can inform therapeutic outcomes. The other aspect that is becoming more and more relevant is screening individuals to identify patients who are at high risk of developing the disease. So it was very much with this that we started to work on 1433 with the opinion leaders to investigate along which of these particular tracks would 1433 offer clinical utility. So I'll focus initially on diagnosis and show you some of the preliminary data around why 1433 is useful as a diagnostic marker. So to date, we've looked at north of 1,000 samples, and specifically in RA, over 400 samples. And what we consistently see through our studies is that 1433 aids in diagnosing RA, so it complements current variables in identifying more patients, which is very, very important to clinicians. Before we get into the data, I think it's important to look at how RA is classified based on EULAR and ACR classification criteria. So EULAR is the European League Against Rheumatism, and ACR is the American College of Rheumatology. So they have joint meetings together, the European key opinion leaders as well as the American opinion leaders, and they come together to develop these classification criteria. And I think what's interesting, the last time that these criteria were revised prior to 2010 was in 1987. So there was a long hiatus between 1987 and 2010, and really I think some of the pressures that drove the 2010 classification criteria was that the field was moving or evolving at a very fast rate. And it was with that they also incorporated additional markers into the classification criteria. So today, 40% of the total classification score is based on serological markers like CRP, ESR. So CRP is C-reactive protein, ESR is the erythrocyte sedimentation rate, and then two other markers that are commonly used to classify the disease are RF, so rheumatoid factor, and ACPA, which has really recently been renamed, and that stands for anti-cyclic citrullinated protein, which is a form of a post-translational modification. So the body mounts an immune response to these citrullinated moieties. And so RF and ACPA are quite commonly used to diagnose RA. CRP and ESR are used more for monitoring and looking at disease activity. I think what's really interesting is if you look at what's noted on the right-hand side of the slide, approximately 40% of RA patients have normal serological measurements. So there's a real unmet need to have additional markers that can complement the current variables to identify more patients and to also assist with monitoring, their disease. And as we go through the presentation, you'll see why it's so important to also have markers that can monitor disease severity and prognosis. So the field, as I was saying, recognizes that there's a real importance in early diagnosis. Not unlike cancer, when you identify or catch a patient very early on, it equates with better outcomes. So they refer to it in the field of rheumatology as the therapeutic window. So the field says that if you catch a patient within the first 12 weeks, there's a better likelihood that the patient will do better, 5 years out. And so really, what clinicians are trying to do is to identify the patients much earlier on, identify who's at risk of aggressive disease so that they can treat them more aggressively. So it was very much with that we started to look at 1433 ADA in the context of RA as a diagnostic marker. So we used established RA, which represents the 135 patients to identify or to establish our cutoff for 14-3 positivity. And so we've looked at various controls that are typically used for a FDA 510K submission or for CE marking or Health Canada approval. And so it was, as you can see from the data on the left-hand side, where we're looking at 135 RA patients compared to the controls, the marker is significantly higher in RA compared to all other controls. And when you run a ROC curve, which is presented on the right-hand side, so a ROC curve is the receiver-operator characteristic curve that allows us to identify the most optimal cutoff, looking at sensitivity and specificity of the marker. So with the specificity of the marker, how specific is that marker for RA? And the sensitivity is obviously how many RA patients do you capture. And so typically speaking, when you establish the optimal cutoff, you want to look at the likelihood ratios for positivity. So we elected to go with a likelihood ratio of 5 as our cutoff based on the advice that the opinion leaders provided to us. And so looking at RA and comparing it to the controls, we realized that the data showed us, that the best cutoff was 0.19 nanograms per mil. So if you have levels of 0.19 nanograms per mil, you're 5 times more likely to have RA than any other disease. So the scale on the left-hand side is 14-3 serum levels, and it's represented as nanogram per mil. I apologize that the descriptor was deleted from the slide. But just know that the data there, the range 0 to 20 is the 14-3 concentration, and it's represented as nanograms per mil. Now, when we move over to the right-hand image of the scatter dot plot, we looked at early RA versus OA. So many times when a patient enters primary care, the clinician may have some difficulty distinguishing between OA, osteoarthritis, versus early RA, because many of the symptoms are very similar. So we elected to start looking at 14-3 ADA in our early RA and comparing it to OA as the corresponding disease control. And what we saw in early RA, in the first 37 patients that we looked at, was that 14-3 ADA is significantly higher in early RA than in OA. So this marker holds promises in assisting clinicians in differentiating between OA versus early RA, which is obviously very important, because if you can catch the patient very early on in primary care and refer them to a rheumatologist early on, then you have a better chance of impacting patient outcomes. And so using the 0.19 cutoff, we've identified that approximately 60% of all early RA patients are positive for 14-3. And we have data now on over 250 early RA patients that shows that 14-3 is a useful diagnostic tool and complements current variables. So I think based on this data, we believe that 14-3 will hold promise in assisting and complementing current variables to identify more patients earlier on. So one of the questions that we received was, is 14-3 ADA, when it exists in the serum, is it there as a bystander of disease, or does it possibly contribute to the joint damage process? And so knowing what the serum levels of 14-3 were in RA, we decided to investigate the mechanism of action for 14-3. So we used, for all of our studies, clinically relevant concentrations to perform cell-based stimulations. And so we've looked at immortalized cell lines, we've looked at primary cells from healthy donors, so PBMCs, and we consistently see that 14-3 is able to induce various factors that contribute to the joint damage process. And now when we look at the in vitro data and compare it to the clinical data, we see that there is a very strong correlation between the in vitro findings and the clinical findings, providing strong evidence that 14-3 is a new target and that it warrants further efforts to try to develop a small molecule, or pardon me, a large molecule to this particular entity. So this is some of the preliminary results that we have, and in the interest of time, I haven't had the opportunity to present everything, but we've looked at various intracellular signaling factors that are relevant in RA. And so what we're presenting here is data on MAP kinase, so the ERK pathway. And one of the reasons why we elected to look at the ERK pathway was that ERK, or MAP kinase, has been shown to be upregulated in the synovium of patients with RA very early on. So there's evidence in the literature that shows that both ERK and JNK seem to be active in early disease and not P38. There's also evidence that when you target or use inhibitors to MEK1, which is the upstream activator of ERK, that you can actually block or inhibit the onset of the disease using animal models. So there's 2 animal models, and in particular, for us, the CIA model is very relevant, the collagen-induced arthritic model of arthritis. So when you look at the data that's presented here, one of the things that we wanted to do was do head-to-head studies looking at 1433 versus TNF as a soluble ligand. And using similar concentrations, we found that in a time-dependent manner, 1433-eta was able to activate or induce the phosphorylation of both MEK as well as ERK. So if you look at the data here, you can see by 5 minutes that stimulation with 1433 leads to an induction or increased phosphorylation of ERK as well as MEK when you compare it to the corresponding control sample. And if you look at TNF as well, comparing again the control to the stimulated, you can see that by 5 and 10 minutes respectively, that TNF alpha is also able to induce the phosphorylation of both MEK and ERK, which equate with activation of these particular targets. And the bottom panel there, the pan-ERK, is just represented to show that equal amounts of protein have been loaded across all conditions. So we've looked now at north of 30 different intracellular kinases, and we've been able to, observe that 1433 exhibits some similarities to TNF alpha in terms of the pathways that are activated, but there are distinct features as well. So, for instance, TNF alpha is known to be a potent stimulator of both P38 MAP kinase and NF-kappa B. The data that we have shows that 1433 actually does not hit these particular targets, so we don't see activation of P38 or NF-kappa B when cells are stimulated with 1433. So we think that because of this, because there's some discreteness in terms of how 1433 signals in, that again, when we're thinking of targeting 1433 with a large molecule, that there obviously will be different outcomes as a consequence of the intracellular factors that they stimulate. One of the next things that we elected to do was to look at, okay, you have all these signals coming into the cell. Now, do the activation of these pathways culminate or lead to the induction of factors that are relevant in the joint damage process? And so when we, again, did head-to-head studies looking at 1433 versus TNF alpha, we saw that, again, comparing it to control on the left-hand side, we saw that both 1433 and TNF alpha could induce factors that are relevant to RA. So we saw an induction of IL-1 beta, IL-8, MCP1, MIP1, TNF alpha, and again, this is limited data. We've explored this in greater context, and we see that 1433 can induce various factors that are intimately linked to the joint damage or the RA physiologic or pathologic process, pardon me. So IL-1, there's actually a drug on the market that targets that. There are drugs under investigation for MCP1, and as I'm sure you can appreciate, The anti-TNFs need no introduction in terms of their utility in the field of RA. There's, I think today, over 5 different molecules, that specific biologic molecules that target TNF-alpha and are used essentially almost first or second line in terms of treating patients with RA. So based on all the data that we have around the mechanism of action for 1433, this is what we believe the mechanism is for 1433 when it exists extracellularly in RA. So on the left-hand slide, just to remind you, the 1433 proteins, and specifically in RA 1433-eta, exists intracellularly. So if we look at this image on the left-hand side, the gray bar represents the cell membrane. So under normal circumstances, 1433-eta would exist intracellularly, and when it hetero or homodimerizes, it binds to various ligands, as I've noted in the earlier slides of the presentation, and through those interactions, 1433 is able to regulate a variety of different physiologic processes, ranging from cell cycle, angiogenesis, differentiation. And I think one of the things that's of interest to us as well is that 1433 binds to the glucocorticoid receptor. Now, your body produces glucocorticoids, which essentially are steroids that when they come into the cell, they would elicit an anti-inflammatory effect. The glucocorticoid receptor needs 1433 to be stabilized. If there's not an interaction between the glucocorticoid receptor and 1433, the glucocorticoid receptor is sent to the proteasome, where it is essentially ubiquitinated and subsequently degraded. When 1433 interacts with the glucocorticoid receptor, it stabilizes it, preventing it from being ubiquitinated, allowing it to bind to endogenous steroid, move into the cell membrane, and to elicit an anti-inflammatory effect. So on the inside of the cell, it appears that 1433 exists and assists in eliciting an anti-inflammatory effect. And incidentally, there are also, when you look at the 1433-eta promoter, there are GR response elements within the actual promoter of 1433. So there is a direct link there and a regulation of 1433 when glucocorticoids essentially bind to the glucocorticoid receptor. Now, if we move over to the right-hand side of the image, in RA, 1433, which normally exists intracellularly, is released into the synovial fluid within the joint. And we believe that that's occurring by one of 2 mechanisms. It's either being secreted directly or it's being released through exosomes. So 1433-eta and the other 1433 isoforms are known to be quite commonly present within exosomes. Now, when 1433 exists extracellularly, as I've said, we've investigated a lot of the mechanism of action for 1433 using clinically relevant concentrations. We see that 1433 is able to induce various members of the MAP kinase family, so ERK and JNK. It also induces AKT, which provides an anti-apoptotic signal. Interestingly, it also activates the JAK-STAT pathway, which is a very important inflammatory driver, and it also activates specific isoforms of the PKC family members, so protein kinase C. And based on this, those signals culminate, as I said, at the level of the nucleus to induce factors that drive both inflammation and joint damage. So when we look at the clinical data, knowing what the mechanism of action is for 1433, one of the things that we've looked at is if 1433 is driving joint damage factors, then we would expect that patients who have more 1433 would have more joint damage. We would also expect that if you have lower levels of 1433, you might do better clinically on certain classes of therapy than if you have higher levels of 1433. So a lot of our additional studies have focused on looking at the relevance of 1433 in relationship to joint damage and with responsive therapy. Now, when we embarked on this program, the initial biomarker, as you know, was 1433-eta. Because 1433-eta exists intracellularly under normal circumstances, one of the questions that arose was, well, is 14-3 eta, seen as being foreign, and does the body mount an immune response to the protein in an effort to clear it? That was one of the first questions that we received. One of the additional questions was, because citronellation is such an active process in RA, and because many of these enzymes that are involved in citronellating proteins exist extracellularly, is 14-3 also a citronellation target? So we've now spent efforts looking at autoantibodies to 14-3, as well as looking at whether or not 14-3 is citronellated. And so when we look at 14-3, initially licensing in a single technology, this has morphed into 5 separate technologies, and it's created a personalized medicine portfolio for Augurex. So on the upper left-hand quadrant, we have 14-3 eta, the protein, which I've been talking about thus far. As I said, in RA, there's these enzymes called peptidylarginine deaminases, which citronellate various proteins, and the body actually mounts an immune response to these citronellated proteins, and hence you can measure, these cyclic citronellated antibodies, which are used diagnostically. And so there, we've seen that 14-3 is now citronellated. We also have seen that as you move down to the lower hand portion of the slide on the left, we've seen that there's specific autoantibodies that are raised to the protein, but there are also autoantibodies that are there that recognize the citronellated form of 14-3. So there's 4 different biomarkers now that we're currently advancing: the 14-3 protein, the citronellated 14-3 protein, and the 2 corresponding autoantibodies. And through our mechanism of action studies, we also believe that 14-3 represents a new therapeutic target, and so that's the 5th technology that we're currently advancing in the field. I'm going to present data today a little bit on the 14-3 autoantibodies, the image on the lower left-hand side of the slide here, and we're currently advancing the 2 citronellated assays, and actually we're just finishing up some studies right now looking at the citronellated autoantibodies and how they complement current variables, as well as the 2 14-3 markers, the 14-3 protein marker and the 14-3 autoantibodies, and the data looks very exciting. So I look forward to the opportunity to share more of that data with you. So when we think of the 14-3 biomarkers and their clinical applications, when we first started off, we were talking about the 14-3 protein and looking along screening, diagnosis, prognosis, and therapy monitoring. We are now looking at all 4 markers along this particular continuum. And the reason why 14-3 eta is highlighted in gray, or a darker shade of gray, is that this test is now commercially available in the US. And as I was saying, when we started off on the call, it's also received European approval and Health Canada approval, and we're just currently working to with distributors to look at how we're going to commercialize the particular test in the rest of the world. So one of the things now that I'll move forward with is looking at 14-3 in terms of prognosis, therapy monitoring, and I'll just present some preliminary data on the anti-14-3 autoantibodies. What's interesting is that when we look at the 2 markers on the left-hand side, the 14-3 eta and anti-14-3 eta, we see that they're very complementary to one another, and that they can be used to stratify patients and identify higher risk patients. So moving along, one of the things that we've elected to do was look at how 14-3 tracks with disease severity, and if it equates with worse outcomes. So what we've seen is that 14-3 positivity and titers, so if you're positive for 14-3, and if you have certain levels, that corresponds with more severe disease, a higher likelihood of joint damage, and joint damage progression. And we've seen this now in north of 300 patients consistently. So we think that that holds promise in terms of assisting clinicians, not only in terms of diagnosing patients, early on, but also assisting in stratification and identifying patients who are at more risk for developing more severe disease. So why is identifying patients with high risk obviously important? Well, if you identify a subset of patients that are more at risk, you would arguably treat them more aggressively, hoping to mitigate disease progression. Now, when we look at the current variables that clinicians have, you can see if the pie chart there represents the total amount of joint damage or predicting joint damage, you can see that the current markers that clinicians have, while they're useful, they don't predict a lot of the variance associated with joint damage. So in fact, when you look at it, it predicts, the current variables predict about 32% of the total variation in joint damage. So 68% of joint damage progression is unexplained by the current markers. So there's a real need to have complementary markers that can really augment the current markers and assist clinicians in identifying patients who are at more risk of developing RA. And so this is actually a really significant effort within the field of rheumatology. And there's a group called OMERAC, that was established to identify markers that can complement the current variables and fill that 68% gap. And right now, they're conducting a study called BioDam, which is to identify biomarkers of damage. And the 14-3 biomarker suite or panel has been prioritized by the investigators to be looked at in this particular formal construct to see if the data that we currently have will hold true in a larger study. So they're recruiting over 600 patients, really with the view of identifying markers that can assist in terms of identifying patients at baseline who are more at risk, but also in terms of the continuum of how patients are treated. They believe that the change in the markers should reflect the change in therapeutic in patient outcomes as well. So as you can see, to summarize from the slide, there's a need for complementary markers that can assist clinicians in stratifying patients who are more at risk of developing more aggressive disease. So when we look at 14-3 eta in the context of disease severity, when we stratify patients at baseline, so when we measure 14-3 levels at baseline before they go on a particular therapy, we see that patients who are positive for 14-3 have higher clinical and serological scores. So the image here, when we look at it, ACPA is anti-CCP, CRP is C-reactive protein, ESR is the erythrocyte sedimentation rate, and HAQ is a clinical outcome referred to as the Health Assessment Questionnaire. So you can see that, for instance, when you look at ACPA, patients who are positive for 14-3 have significantly higher ACPA, they have higher CRP compared to the negative patients, they have higher ESR, and they also have significantly higher HAQ. So at baseline, 14-3 identifies patients who have worse disease. So if you're 14-3 positive, you would expect that they would have more severe disease. When we look at this now and look at longitudinal data, which is reflective of the image on the right, the line graph there, we see that, again, patients who are positive for 14-3, when you look at radiographic outcomes, so how much joint damage do they actually have within their joints, we see that the rate of radiographic progression or the burden of radiographic damage is higher in patients who are 14-3 positive compared to 14-3 negative patients. So arguably, if you're 14-3 positive, you're more likely to progress and develop more severe disease. Now, one of the things that's really of interest for 14-3 in those seronegative patients, so the patients that are 14-3 negative, you can see that some patients do progress. Right now, we've only looked at 14-3 eta levels at baseline, so at time 0. There are ongoing studies that will look at 14-3 eta levels over the 5 years in those seronegative patients to see if some of those seronegative patients convert and actually become seropositive for 14-3. And we feel that there's value in routine measurement of 14-3 because, when we look at the other markers, there is some evidence that some patients will seroconvert and become positive. So we do believe that 14-3 as well. And we do have data that suggests this in small cohorts, that patients do have the opportunity or the ability to seroconvert and become positive for 14-3. So for us, it would be very interesting to look at the patients who were initially seronegative, and especially in those patients that develop more radiographic progression. Over time, do they become seropositive for 14-3? But the data as it stands right now to date shows that if you're positive for 14-3 at baseline, so time 0, you're more likely to have more severe disease and more likely to develop more aggressive disease as marked by radiographic damage progression. So when we think back to the in vitro data showing that 14-3 ADA induces factors that are intimately linked to both inflammation and joint damage, clearly, the clinical biomarker data supports the in vitro data in that if you're positive or you have higher levels of 14-3, you're more likely to have more severe disease and develop more joint damage. So there is that alignment between the clinical biomarker data and the in vitro mechanism of action data that further substantiates 14-3 as a rational drug target that should be further investigated. One of the next things that we've looked at is 14-3's relationship to therapeutic outcomes. So if you remember from the mechanism of action slide, we saw that 14-3 ADA was able to induce factors like TNF alpha, IL-1. We also have data showing that it induces IL-6. And so there, actually, and I don't have this data presented there, but we have data in in a dose-dependent fashion showing that when you stimulate cells with 14-3 in a dose-dependent fashion, as you have or increase the concentration of 14-3, you increase the output of these different ligands. So higher levels equate with more induction of these inflammatory factors. So there's a dose-dependent effect. And so when we think of that clinically, that's important because, again, do higher levels of 14-3 equate with different patient outcomes? And so, as you can see from this particular slide here, lower levels of 14-3 at baseline together with a decrease in 14-3, meaning that if you measure 14-3 at baseline and then 15 weeks following therapy, if the levels decrease post-treatment, that equates or corresponds with better response to therapy, as determined by disease activity measurements. And we've seen this now across different classes of therapy, such as DMARDs, so disease-modifying anti-rheumatic drugs like methotrexate, as well as with anti-TNFs. So in the interest of time, I'm going to present data around the anti-TNFs today, but I'm happy if someone would like to get in touch with me after the fact to share data on the DMARDs as well. So why is there a real impetus in the field to find markers that can predict outcomes? Not unlike cancer, obviously, you want to find markers that reflect better patient outcomes to improve patient outcomes, but also to reduce the economic burden of the disease. Because RA is a chronic disease, it's very costly, and payers now are really tightening things up, wanting to have better outcome data, and at the end of the day, what they're really, kind of pushing folks to do is to align any any drugs with markers that can afford, the patient as well as the payers to identify patients who are more likely to respond right at the outset, as opposed to not having something like this where arguably you just follow standard of care. They really want to align any new therapies with the markers so that you can, not unlike what happens in breast cancer, if you have marker X, arguably you treat the patient with drug X or if you have marker Y, you treat the patient with a different drug. So the classic Herceptin HER2 situation is something that's of real importance in the field of rheumatology, really to improve patient outcomes and reduce the economic burden of the disease. So this data represents data in patients who were treated with an anti-TNF therapy. So these are RA patients that were treated with various anti-TNFs. So we're not looking at a specific molecule within that class. We're looking at all molecules within that class, and this is data represented on 74 patients. Before I go into the clinical data, if you just remember from one of the earlier slides, we had shown that 14-3 can actually induce TNF-alpha. We've now looked at TNF-alpha, and we see that TNF-alpha can also induce 14-3. And ourselves and other people now in the literature have shown that there's an interplay between 14-3 and TNF-alpha, in that TNF-alpha can induce 14-3. So based on this interplay, we think this rationalizes why in some patients we might see that when you treat a patient with TNF-alpha, that arguably the levels of 14-3 decrease because of this particular nexus that exists within cells. So moving over now from the RT-PCR data to the information presented on the upper right-hand side, we see that when patients are treated with an anti-TNF, that the 14-3 levels are modifiable. So at baseline, when you measure baseline levels and look at 15 weeks out at the 14-3 levels, we saw that there was a significant difference between 14-3 levels at baseline and those at 15 weeks out. And so with that, if you look at the change in 14-3, what we saw when you begin to look at patients in terms of response, if you look at those patients who achieve a good EULAR response, meaning that they do well clinically, if we look at the change or the decrease in 14-3 and equate it with the change in serological measures in the good EULAR responders, and importantly, the change in ESR, CRP, and DAS are used as routine measurements to monitor patient outcomes, we see that a decrease in 14-3 equates or strongly correlates with a decrease in ESR, CRP, and DAS. And when you look at the R square, or the R value, for instance, just looking at the change in ESR with the change in 14-3, you can be confident that that's a real phenomenon when you see a p-value like that. Now, if we move over to, so essentially, sorry, before I move over to the left-hand side, the change or the decrease in 14-3 is equating with better clinical outcomes. Now, when we move over to the information on the lower left-hand side, this is data generated from a ROC curve, so a receiver-operator characteristic curve. When you look at patients who do well, so achieve a good EULAR response, compared to those patients that do not achieve a good EULAR response, and you look at baseline measurements of 14-3, what we see is lower levels reflect, or lower levels of 14-3 equate with a better clinical outcome. So when you look, for instance, at a cutoff of less than 0.41, you're 5 times more likely to do better or to achieve a good EULAR response than patients who have levels above that particular 14-3 level. So lower levels equate with better clinical outcomes. So you can see that if a patient has less than 0.27 nanograms per mil, they're 8 times more likely to have a good EULAR response. So if we think of that mechanistically, as the levels of 14-3 eta increase in serum, it's going to be driving up more factors. And even though the patient may be having an appreciable benefit to that particular therapy, because 14-3 exists in the background at high levels, is going to be continually stimulating cells to induce these different factors. And that might explain in part why the patients don't do as well clinically. So lower levels to begin with and a post-treatment decrease equate with a higher likelihood of a clinical response. And we've regressed this out through modeling, and that data holds true. So lower levels with a post-treatment decrease predicts who's going to do better. Now, looking at the 14-3 autoantibodies, One of the things that is exciting to us and further validates the importance of developing a therapeutic to 14-3 eta is that what we see is that the autoantibodies complement 14-3 in diagnosis, but that when you're positive for 14-3 autoantibodies, you have more favorable outcomes. So they appear to operate as endogenous therapeutics that protect the patient. So the view there is that if you are 14-3 autoantibody positive and you have higher levels, that you do better clinically. So this is data that we've presented recently at one of the actually at EULAR, just recently in Madrid. And what we see here is that when you look at 14-3 autoantibody levels, and if you look at the scatter dot plots, what you see is that the 14-3 autoantibodies are significantly higher in early disease compared to established disease, where the median and interquartile ranges look comparable, to healthy individuals and patients with OA. So for whatever reason, the autoantibodies seem to be preferentially expressed in early disease, and that when the disease progresses, there's only a subset of patients that are able to sustain this particular autoantibody response. So if you look at established disease, there are a subset of patients that have levels that fall within the range of the early RA patient set. And so when we look at this is quite exciting because of the fact that these autoantibodies are very high in early disease. Does it create an opportunity to capture that 40% of patients that may be seronegative for the other markers such as anti-CCP and RF? And if so, does it complement 14-3 as well in terms of diagnosing patients? So when we look at the receiver operator characteristic curves, not surprisingly, we see that the 14-3 autoantibodies are highly specific for early RA compared to healthy individuals, OA, as well as all controls. So an area under the curve close to 1 represents a very good and perfect test. So you can see from the data there that these autoantibodies with the area are approaching close to 1, and so they're highly significant as denoted by the p-value there. And it's worth noting that this is data based on 550 patients. So again, it's not a small cohort. And we've now expanded the early RA study looking at north of 250 patients, and this data holds true in larger cohorts. Now, I think what's interesting, just quickly going back, is if you look at established disease and those patients that are able to sustain the autoantibody response, so the patients in gold there, when you look at the patients who have high levels and who would be positive for the autoantibodies, what happens to their patient outcomes? And so when we look at a subset of these patients, when we look at patients who are positive only for the protein, so the 14-3 protein, as denoted by the 15 patients there, versus patients that are only positive for the autoantibody, 35 patients. So i.e., they're only positive for one of the markers, either the protein, so they're negative for the autoantibodies, and the autoantibody positive patients are negative for the protein. When you look at their joint damage scores at year 4 and year 5, you see that the patients who are positive for the protein, not positive for the autoantibodies, have higher joint damage scores at year 4 and year 5 than those patients that are only positive for the autoantibodies. So it means that patients who are positive for the autoantibodies seem to be doing better clinically. And we have data around this as well with response to therapy, that if you're positive for the protein and negative for, if you're negative for the protein and positive for the autoantibodies, that you actually do better clinically as well. So we're now embarking on studies to look at this relationship in a broader sense, to look at its complementarity in terms of diagnosing patients as well as prognosticating patients. So can we use these two markers really to subset patients into different risk categories, and can these two markers also assist us in delineating or predicting therapeutic outcomes? So This brings me, and I'm almost close to the last slide here, but priorities for Augurex are, as I was saying, to focus on commercialization of the 14-3 protein test. So it is marketed in the US. We've also received Health Canada and European approval. So we're now embarking on distribution agreements to make 14-3 the protein test broadly accessible. We're also focusing on developing IVD tests for the other three biomarkers. And I'd say that we're around the corner with the anti-14-3 assay, having something that would be ready, or close to go to market. The other two assays, the CIT and anti-CIT assays, are markers that are slowly coming up, and we expect by, I would say, Q2 of 2014 to have those markers available for clinical use. And one of our other priorities, not surprisingly, is to advance the therapeutic program. So one of the things that we're currently doing there is screening models to identify a relevant model that would actually express 14-3 in the autoantibodies in a comparable way to patients. And so we've already started to look at the collagen-induced arthritic model, and we see that upon induction, that the 14-3 protein is actually present, and we can measure it, and that the animals also, within 10 to 12 days, start to develop an autoantibody response to the protein. And so the view there is to use the markers to identify which animals to treat and how to treat the animals. So for instance, if you're mounting a good response on your own to the protein, you might consider dosing that animal differently than an animal that arguably has the protein but does not develop a robust autoantibody response. So we feel that the markers, together with the therapeutic program, represent one of the first personalized medicine strategies for rheumatoid arthritis. So here's some of the abstracts that we've presented on 14-3, and you can access that information also through the Augurex website. And with that, I thank you for your time, and if you require any additional information, please feel free to contact me. I've provided my email address for you, and I thank you for your time. And that's the end of my presentation. Thank you. I'll take any questions that anyone may have. So I thank you again. So I just received a question from Pierre Bouton. The question is, what is the animal model you quoted for RA? Are there any more? Yes. So there's a number of animal models that are commonly used. So there's the collagen-induced arthritic model. There is the KXBN model. There is the AIA model, which is a rat model. There is the CAIA model, which stands for the collagen antibody-induced arthritic model. And if you'd like to reach out to me, I do have a review paper that cites different animal models within the field that are commonly used, and I'd be happy to forward you a link or the actual paper itself. Any other questions at this time?